Today's job is to repeat an analysis using a different software. I use RaxML for so many different types of phylogenetic analysis. It's really exceptional software, but it's unwise to rigidly stick to one approach. Better to keep an eye on other methods, other software, and so that's why today I'm trying IQtree for a particular type of analysis. This is a species comprehensive analysis. It is constrained with a multiplicating constraint tree. It has two partitions, colon barcode and a large set of binary characters for matrix representation of a whole lot of source trees. Now normally I do this in RaxML. So here's the command I would normally use. MPI version. I'd use ignore the weights file for a moment. Constraint tree with a -G switch. File format alignment containing both colon DNA sequences and the binary characters, and a partition file and a bootstrap. So I've just been figuring out how to do this in IQtree, this equivalence analysis. I've just got it working now. You can see it running. Not very much memory at the moment. I assume that will go up a lot when it's onto the following stage. But anyway, so very quickly, what was necessary. Now it seems IQtree is you can use the XML format in a partition file. But it appears that you need to use the Nexus format when you have partitions of different types, which I have. I have the DNA and the binary. So this is a format for specifying your partitions. I actually have to give the name. I should say firstly, you need in RaxML you have a concatenated alignment, whereas in IQtree you have separate alignments. So first it was necessary to separate them out. Now imagine you concatenating and then into RaxML, but you can't exactly use the. I'm not sure you can use the starting files, so I actually split them back. There is a RaxML function -fs for splitting a supermatrix into the individual partitions, and that creates these two files. I'm not sure, maybe may need to remove species with no data, so there'll be a few of those. So these are species with none of the binary constraints. Maybe it's necessary to do this. The other main thing is you need unrooted constraint tree for IQtree, which is different from RaxML. You can use rooted, maybe unrooted also, but apparently IQtree. So I needed to unroot a tree. I checked the two approaches I used for browsing trees, Archaeopteryx and Figtree, and neither of them you could unroot, it appears, at a quick glance. So I've used ape, the ape library. So open R, load the ape library, read the rooted constraint phylogeny, multiplicating, but it doesn't matter. And use the unroot command in R to unroot that, and then just write the tree, and it seems automatically writes it to numeric format. So that's the main thing. Now I think this is IQtree version 1, so you use the SPP to specify the partition file. Oh, fine, go ahead. And then the -G for the constraint file, constraint tree name. number of bootstraps. But again, the main thing was to make the partition file. Now the format is slightly different, it seems, so you have to use this Nexus, as I say, not the VAXML. So I converted the VAXML format, and I made, if you do that, maybe one one silly mistake, well, I made at least, is to retain this numbering. So partition 1 starts at 1, goes to, that's the length of the colon. Then partition 2 is the next character. Now, but because you're specifying 2 different files, you have to actually start from 1 again. So that's a main mistake. Make, and it's not really an error message to alert you to this exactly. It's a bit ambiguous. And this is the error message I think it gave: too large site ID. So that error message is probable from not specifying the character start and end positions correctly. So just need to swap this for this in when specifying the second partition, and then you need to give the models for each partition. So GTR for DNA, GTR2 is apparently for binary or morphological characters, and that's it. That ran okay. Now the reason I started this, as I say, it's a good idea to assess different software. I did recently browse to one tree, and in a supplement there was a VAXML tree and a IQtree result, just those two, and superficial, after just a quick browse, it seemed the IQtree was much better resolved, so that's why I thought I would try this. But that's not really, it's not really a good reason. It's just a generally good approach to try different software for all your different types of inference tasks. So the next thing is main thing is criteria would be how long this takes. Is this horrendously slow? For VAXML, I think this is about a day to do this job, and we'll see if IQ does it does it take longer. I will probably transfer this to a different computer to let it take as long as it takes, and see what is the memory requirement, which is another criteria. Well, we'll see. But otherwise, job done. We are now running an alternative approach, and we will see if that compare the results when we get them in.